So much, and I can honestly say that it's very nice to be back. I think this is about my fifth trip here to Ann Arbor to the university, the first since COVID, so it's really nice to be back. And I really appreciate the invitation to come and present some of our work. I actually sent 2 abstracts and let each department decide which talk, and I was a little bit surprised because I thought, well, I'm coming to the mechanical engineering department, mechanics series, but the talk that was selected was actually one that had a little bit more biology. So I'm going to share that today. I know that not everyone thinks about blood vessels on a daily basis, at least not from a research perspective. So let me use 2 slides just to orient everyone. But I'd like for you to think of these arteries as sort of an archetype of soft tissues and how cells influence soft tissue structure and function. So what you see here on the left is a micro CT image of the central vasculature in the mouse that we have now rendered into a computational model. And I thought this would be a good first slide to show because this was rendered here at Michigan by my colleague Alberto Figueroa. If you look at these sections, these cross sections on the upper part of the slide, we use different stains, histological stains, to identify the different constituents that make up these tissues. And if you look at the stain on the left, we can then, using some software, just extract out the different colors. And so this, in the bottom left, shows the elastic lamellar structure, which is really characteristic of large arteries, and in particular, the aorta, which is the largest artery in our bodies. If you look at this image, what we see in blue is collagen. And collagen is the most abundant protein in our bodies. It's found also in arteries. The collagen will endow the wall with stiffness and strength, whereas the elastin endows the wall with its truly elasticity, its ability to distend during systole and then to recoil during diastole. And then you see on the bottom right, smooth muscle cells that occupy this middle layer, which we refer to as the media, and the outer layer, the adventitia, primarily collagen. So that's just for orientation. We're going to talk about arteries today. So you see here a video microscope capture image from our ex vivo test system. So we'll take these vessels, excise them at various times, cannulate them, place them in really a biaxial bioreactor. And I will talk about the descending thoracic aorta primarily, but in the latter part of the talk, also the infrarenal abdominal aorta. So those are the 2 segments we'll be interested in. Physiologists want to know, when you pressurize a vessel, how much will it distend? We would also like to know, if we apply an axial force, how much will it elongate? And as engineers, we want to turn that into stress strain data, which is shown on the right in the circumferential and axial directions. Conceptually, the area under stress strain curve represents the energy stored. There is some dissipation, so we always look at the unloading curve, and that tells us how much energy this vessel would store, not dissipate, and be able to use to work on the blood to augment flow. And conceptually, these tangents would show the stiffness, again, biaxially. And I say conceptually because we actually use a theory called small deformation superimposed on large in order to calculate the stiffness at various points in the cardiac cycle. So that's by way of background. Now I'm showing in the upper central portion a cross-section of a normal artery, the aorta, the descending thoracic. And now we're going to look at what happens when you elevate blood pressure. Unfortunately, in this country, over 100 million Americans have hypertension, that is, increased blood pressure. And the question is, what does that do to the vasculature? You may not be able to appreciate it, but this hypertensive wall has thickened some. Now, the 2 simplest equations in arterial mechanics that come from mechanical engineering, the first one from the fluids. So if we were to solve the Navier-Poisson equation, or really the Navier-Poisson equation for the constituent equation, introduce that in linear momentum balance, have Navier-Stokes, solve that in a cylindrical tube, fully developed, steady, laminar, we can compute the shear stress that would be exerted on that surface. And that's shown here, where mu is viscosity, Q is the volumetric flow rate, and A is the inner radius. it turns out that this shear stress is regulated by the cells to remain nearly constant throughout life. So let us introduce a parameter epsilon that is the fold increase in cardiac output. So if you were to go running, your heart would increase its output, so that's cardiac output, and let's say it increases 10%, epsilon would be 1.1. So we're going to increase this flow by this fold change epsilon. We introduce it here, and you might notice that there are some subscripts O for original. So these are original values of flow and radius. These are current values. And the question is, how would the vessel adapt if you increase cardiac output in order to maintain the shear stress constant? And the answer is the radius would have to increase or decrease according to this rule. Now, in hypertension, cardiac output tends to be maintained, so epsilon is nearly 1. The second simplest equation in vascular mechanics is the Laplace equation, which mechanical engineers, you learn this in strength materials courses. This is the hoop stress, which is determined through the descending pressure P, the radius, and the wall thickness. Again, subscript O says this is the original value. This is the preferred value, the target value. What if we increase pressure by gamma fold? What this rule says is in order to keep that stress constant, the cells will have to produce matrix. They will have to thicken the wall according to this rule. If we look at this vessel and check these two equations, we find that we can check off that indeed these cells have responded to this elevated pressure in a mechanobiological fashion, and they have remodeled the wall to maintain these target values. You notice in the bottom left, I have 9 out of 11. So we looked at 11 mice, and 9 looks, this is represented the 9. You don't have to be a vascular pathologist to know that these two are different. So this is, my subtitle is A Tale of Two Mice. So the question is, what happened in these two cases? Now, when we went back and looked through our records, it turns out that these two mice were from the same litter. They had the same parents. So we thought there was something genetically different, a so-called genetic modifier. And this would be important to find. Now, another thing I didn't mention is when you work with mice, we often work with pure or inbred strains. And one common strain is called the C57. Another is called the 129 mouse. These mice were actually a mixed background, which had these two genetic backgrounds, and these mice are commonly used to develop mutations in mice. And we study we've studied over 50 different mutations in the lab. So we had two options now. We thought the hypothesis was that maybe in one case, maybe the 129 background was dominating. Or maybe the C57 was dominating. So we could have taken these mice and backbred them many generations to pure backgrounds, which would have taken months. Or we could simply call the vendor and buy pure C57 and 129 mice, and that's what we did. Now, it's common these days in biology to do RNA sequencing, which is to say to look at what genes are being expressed by the cells of interest. And in many cases, these so-called differentially expressed genes are representing what's called a volcano plot. And every dot shows a gene that's different between these two backgrounds. Now, many of these have unknown significance. But if you go through and do a so-called ontology study, you find out that one of the differences seems to relate to the extracellular matrix. That is the scaffold in which the cells of the wall reside. So we want to look at that matrix. But if you look at gross measures of the medial elastin, that was the black lines in that bottom left curve, figure that we saw, or perhaps the collagen or the smooth muscle cells, or something I didn't mention, so-called glycosaminoglycans. These hydrate the wall. There are some slight differences, but none of them were statistically significant. And indeed, if we looked at blood pressure and the stress in the wall at blood pressure, the stiffness, and this bottom row says, if we gave an agonist to the vessels, they have muscles, so they can contract. How much could they contract? And they could reduce the diameter by anywhere between 19 and 23%. None of these were statistically significant. So we didn't yet know what was maybe going on. So in the first case, we made the mice hypertensive using one method. We thought, well, let's try a separate independent method and see if we also get a response. And again, you don't have to be a vascular expert to know that there's a difference. So this is the C57 aorta, this is the 129 aorta, two genetic backgrounds. We give a potent vasoconstrictor, a biomolecule called angiotensin II. And what we see is this is the thickening of the C57, and this is the thickening of the 129. So we now had a clue that those two mice probably had some dominance somehow during their breeding, had some dominance of the C57 background. And this just quantifies the different constituents in the wall, and you can see how thick the wall is. This is cross-sectional area. This outer portion we called adventitia has increased tremendously. This is mainly collagen, but even the media increased, both through additional smooth muscle and collagen. So now let's look at the so-called RNA data, the bulk RNA sequencing, when we now make these mice hypertensive. And we see that there are many, again, differentially expressed genes. But now we have a little bit of insight. And so what you see here are 16 columns. The first four are the C57 normal, then the 129 normal, then the C57 hypertensive after 2 weeks, and the 129 hypertensive. And this is just listing a few of the many genes. This first grouping has to do with contractility. And what you see as the color goes from this orangish to this blue, there's a decrease in contractility in the C57. We have our first clue of something being different. We look at the second group and we see this bright red. This means an increase in the extracellular matrix that's being produced by the cells in those aortas. Second clue. And then finally, this one band, this real bright red relative to the basal level here, this is a so-called cytokine. It's an inflammatory cytokine called interleukin-6. So this suggests that maybe inflammation is involved, contractility is involved, and it's somehow affecting the ability of these cells or the response of the cells in making extracellular matrix. All in response to what? We're in mechanical engineering. High blood pressure, which is increasing the stress on the wall. Nice thing about bulk sequencing is it can be quantified. The other nice thing is that you can run software that basically looks at groups of genes and says, this grouping relates to, I've pseudo-colored here in green, contractile processes. And in the bottom group, pseudo-colored in red, genes, groups of genes that relate to inflammation. So it sounds like maybe a difference between these two responses, this tale of two mice, in hypertension, the same elevation of blood pressure, the same initial elevation in wall stress, that smooth muscle contractility, inflammation are playing a role. So this told us to go back and look, and I don't know, is that, yeah, it's showing up better on your screen than mine. So here's the C57, the 129, normotensive, hypertensive, and what we've stained with is a stain that looks for all leukocytes, that is white blood cells. They're part of the immune response, and living through COVID, we know all about immunity, right? And so T cells, some of your adaptive cells, they would light up here as brown. And what you see is an increase in the middle layer, but this dramatic increase in this outer layer in these white blood cells. And remember this rule that if the thickness increases proportional to this fold increase in blood flow to the 1/3 power, blood pressure to the 1 power, that if this rule is obeyed, we say it's adaptive. We use the word homeostasis. The Greek homeo means similar to, stasis literally means standing still. Mechanical engineering, we talk about statics, right? So standing still. So what we see is that if we plot this nice metric wall thickness as a function of the degree of inflammation, we get a near linear response. So the more inflammation, the thicker the wall, and the loss of this adaptivity. So inflammation is driving something, in this case, that's not adaptive. Well, inflammation and what? Contractility. So these plots, this is diameter on the ordinate and time on the abscissa. And so at time 0, the blood vessel has unit 1 diameter. And then we give various drugs and or chemicals to make the smooth muscle contract. This is ex vivo. This is in a well-controlled, computer-controlled environment. And you see the vessels will contract down, reduce their diameter. The one on the bottom, that's the 129. Next, we gave angiotensin II, not chronically to develop high blood pressure, but transiently ex vivo to see how responsive were the cells to this agonist. And again, the 129 was most responsive. We can skip these and go right to this plot. And now you see, it's plotted a little bit different. So this is 0 contractility. This is full contractility. So the more contractile the wall, the less thickening. That is, the more adaptive. And that's really what that muscle is there for, is to contract and what we call vasoregulate. So I mentioned this Laplace equation before. It's very simple and yet intuitive. Distending pressure, luminal radius, and wall thickness. But when we think of radius, we really have to remember that radius depends on what? The distending pressure. You pressurize the wall and it will achieve a larger radius, and that's shown here. So the radius goes up as we pressurize. But as we contract the wall, the muscles will work against that pressure and actually reduce A. The wall will thicken as it contracts and thin as it as you pressurize it. So the stress depends on not just the mechanical loads, but also the activity of these smooth muscle cells. And now we know that activity is different in the 2 mice. So now we're going to look at a very simple plot of the stresses in the circumferential direction, not using Laplace, but using equations from nonlinear elasticity to solve the stresses in a thick-walled cylinder that's bilayered. Each layer has separate nonlinear anisotropic properties. Okay, so this is something you'd learn in continuum mechanics or finite elasticity. And what we find is that the stresses in that medial or middle layer are much higher than the stresses in the adventitial layer. And this makes sense teleologically. It makes sense from a biological design perspective. Do you remember where those black lines were? Where that elastin was? It was in the middle layer, the media. And what you want is for that wall to distend and store energy in these elastic structures during systole. That energy can be used during diastole to recoil the wall, to work against the blood, and essentially squeeze it. And that augments flow, antegrade and retrograde. So you want the stress to be borne by that middle layer. What is the collagen, which really populates that outer? What did I say the collagen does? Stiffness and strength. It's really a protective sheath. So I don't do this anymore, but if you were to go and lift a lot of weights, and your blood pressure would go up acutely, that vessel would distend, but that collagen would engage and lock and prevent it from over-distending and rupturing. So this is a protective sheath that's engaged when blood pressure goes up. So you don't want that protective sheath to be engaged under physiologic loads. Well, now let's see what happens if we pressurize the system. The stresses go up in the media. That makes sense. We have a thick-walled tube that we're pressurizing. But notice that the stress increase in the media is much less than the stress increase in that protective sheath. So it's starting to engage, it's starting to do what it should do. And we've increased the pressure acutely, consistent with the level of hypertension. But it's not just this passive wall, they're also smooth muscle cells. So they can contract against that pressure. And so they can bring the stresses back down. And what we noticed is, and it's a little bit light here. Again, it's showing up fortunately better on your screen than mine. This is the 129 adventitia. Now I've put in this pseudo-colored line here to show that despite pressurizing the vessel to this hypertensive level, because these smooth muscle cells can contract against this angiotensin II stimulus, they can return the stresses back toward normal in the adventitia. But the C57 mice have compromised contractility, so they can't. So this stress is higher than it should be. So this is where the mechanobiology comes in. These cells are sensing a dramatic increase in stress, and they're going to respond to that through gene expression, probably to make matrix, to thicken the wall, to reduce the stress, okay? So when you go and you exercise and you do lift some weights, what happens? You build up muscle mass to offset this increased load that your muscles are used to experiencing. But this is just acutely. This would be within minutes. We're really interested in what happens over a period of weeks. And we can go through and so each of these panels, these 6 panels, are individual finite elasticity solutions of a bilayered wall. Each layer has a separate nonlinear anisotropic material, and the middle layer also has smooth muscle contractility. But what you see is that this adventitial stress, which was originally nearly the same in the C57 and 129, is preserved even after weeks of hypertension in the 129. It was able to adapt. We call this a homeostatic response. It can respond to the perturbation and load and maintain nearly normal the stress. Whereas you see the C57, the stress is not higher than normal, it's actually lower than normal. It has over-responded, it has maladapted, and we'll look at the reason for that. Mechanical homeostasis is this concept that there are target values, set values that are preferred. And the cells will always sense their mechanical environment. They're literally reaching out and assessing their mechanical environment. And if the environment, that is the state, which they are sensing, is consistent with the set point or target value, which means the deviation between the sense state and the set point is low, it's within a tolerance, then this is just normal physiologic turnover of matrix. But what if the sense state is different from the set point? What if stress goes up? What if stress goes up because pressure goes up? Then the cells will sense that, and they will, through this system, which is controlled or defined by gains and rates, work in a negative feedback fashion to try to return that state toward normal. That's homeostasis. We'll take as our state, the state of stress. And this sigma is a scalar metric. So this is actually the first invariant of the Cauchy stress tensor. We've introduced a parameter delta. So if delta is 0, it says the cells can sense exactly what their environment is. And as delta goes towards 1, it says they're losing their ability to sense their environment, which means they're losing their ability to effectively respond to that environment. So we take as our deviation this difference as the difference between the sensed state, the target value, and we normalize it so it's a non-dimensional parameter. Well, shown on the bottom is just schematically homeostasis. So this is stress. So today we have all walked. We've walked upstairs, we've walked downstairs, we've stood up, we sat down, we ate, etc. So all during the day, blood pressure is doing this. But the vasculature is responding to that and making sure these quantities are maintained. Even if you ran today, maybe to catch a bus, this was maintained. There are other cases over longer time scales where the stress is perturbed, like in hypertension. And a homeostatic system will work to restore that stress back toward normal. That was our 129 mouse. So what we want to do is put into a continuum formulation this idea of mechanical homeostasis. And what we chose to do is to use something that we called a constrained mixture model. And this was originated or conceptualized with Professor Rajagopal at Texas A&M and myself. And essentially what we said is we will try to satisfy mass balance using a full mixture approach where constituent alpha will represent elastin and collagen and proteoglycans in cells, etc. But we're going to just satisfy linear momentum balance, F equals ma, using a traditional continuum descriptor. So mixture theory on mass, normal continuum mechanics on linear momentum, We know from the second law and definitions of the Cauchy stress written in terms of the deformation gradient F and C, which is F transpose F, that if we have a potential function here called a stored energy, that we can compute stress, put that into linear momentum balance and satisfy equilibrium, assuming this is a quasi-static process. We're looking over 2 weeks and things are happening slowly. It's like watching grass grow. Over 2 weeks, it grows. But on short time scales, you don't see a change. And to complete this formulation, we're going to use a rule of mixture so that the total stored energy will be the sum of the stored energy in the constituent parts. So if we think of homeostasis, you can imagine a configuration, or if you will, a neighborhood about a point, and over time, there's no change in that configuration. But even though there's no change, right now, for all of you young people, graduate students, et cetera, I have some bad news. The collagen in your aorta is degrading. Remember, it provides stiffness and strength. But I also have some good news. It's being produced at the same rate, at a rate that balances exactly the degradation. So it's offsetting. So even in unchanging mechanical states, there's yet this dynamic process where cells are making and removing, okay? And when all that happens in an unchanging state, we call that tissue maintenance. Now, as the cells are making this material continuously, they pre-stress it. They put it into the existing matrix under tension. And they do that through actin-myosin activity. And we introduce this G, which is a deposition tensor. And so under homeostatic conditions, this defines the stretch or the deformation gradient in 3D that the individual constituents will experience when placed within the material. And so we postulated, this is a postulate, this is a constitutive postulate for the energy stored by constituent alpha at any time. And it really has these three key contributions: the rate of production, the removal, which really quantifies, we know all biological tissue has a finite half-life, and then this mass averaged energy. So why do we pick that constitutive assumption being this? Well, imagine at time 0. So 0 is homeostatic, everything is normal, you haven't had surgery, you're not hypertensive, you haven't started training for a marathon, is time 0, everything's normal. So S is a growth remodeling time. When S is 0, the integral goes away. This Q tells us how much of what was present at time 0 is still there. So at time 0, it's all there, so that's 1. And so we look at time 0, we have the apparent mass density of each constituent. This is essentially a 1. We divide by the total or mixture mass density, and what we end up with is a simple rule of mixtures as we desired, where phi is a volume fraction. More interesting case is over all these growth and remodeling times, what if, as we looked here, what if that state is not changing? Then this deformation gradient is not changing. It's referred to a configuration, a so-called natural configuration that's the same. So this W can come out of this integral because it's a constant, and what you find left in these parentheses is nothing but that apparent mass density. So again, you divide by the mixture mass density, you get your volume fraction here, and or mass fraction here, and you get your mixture. So you recover the special cases that you want. Is the constitutive postulate correct? Probably not. Does it satisfy special cases? It does. Is it worth trying? You bet. So we tried it. So now the key thing comes down to, and I've had so many wonderful conversations throughout the day, and I forget which conversation it was, but it often comes down to the constitutive equation. And now we have 3. What dictates the cell's production of matrix and how that change? What dictates the half-life of whatever's being produced, and what are the properties? This is actually the easiest, which is usually not the case. So it may be a new Hookean descriptor or a Fung exponential or Mooney-Rivlin. But let's look at production removal. This is the basal production rate. Remember, all of you students right now, fortunately, there's material that's being made that's offsetting the material that's being lost. So there's this dynamic turnover, which for decades in periods of health is this perfect balance. It's remarkable to me. But when the stress is perturbed from normal, that is pressure driven, or the shear stress, which is flow driven, is perturbed from normal, the rate of production will change. And how sensitive it is to that change is dictated by these gains. Remember the gain in that system? Just like a control system, you have gains. So these are gain-like parameters. We'll come back to this one momentarily. And the same thing about the half-life. This is a generalized first-order kinetic decay, but this basal turnover rate, or this is related to the half-life, can also be modulated by changes in the mechanical environment. So conceptually, we looked at tissue homeostasis, and we looked at homeostasis in the case of perturbation. I'd like to now introduce this, and this was not an idea that I had. This actually comes from Ruslan Mezatov, who's an immunobiologist at Yale. So you imagine there's a homeostatic state. This is the set point, and there's a range. There's a tolerance. And as you change loading within that, you're not going to have a dramatic response. You're going to be able to get back to that homeostatic set point. There can be cases where there are perturbations that are larger, so your deviation from that set point is greater, but there's a stress response and it's homeostatic. And this is remarkable. Our vasculature and other tissues in the body are incredibly adaptive. And this is sort of conceptualizing that. Now, there's another case where it's adaptive, but what you see is that you don't recover the set point. So instead of just homeostatic, we refer to this adaptive homeostasis. And often this occurs with some inflammation playing a role. And so you see, and what Ruslan has coined this as para-inflammation. So if we step back, and now that we're, can I say we're through the COVID period? I don't know if we're, now that we have experience and we've thought a lot about COVID, we know and we are very thankful for the fact that our immune system essentially evolved to protect against threats, bacterial, viral, fungal, et cetera. And these can be life-threatening, as we now know. So this is a prioritized system. And prioritized systems take precedence over other systems like mechanical homeostasis. Mechanical homeostasis helps to preserve optimal function, but not necessarily life. The immune system is there to protect us in cases of deadly threats. So a prioritized system will win. And there will be cases where inflammation may play such a role that you go beyond this homeostatic response, beyond this adaptive response. You survive, but it may not be optimal. And so schematically, you see a dramatic perturbation, and in fact, you go beyond the set point, and this is what we refer to as compromised or lost homeostasis. Does it make sense? So we have all of these different options because we have a highly responsive, mechanoresponsive, and adaptive system, but we also have multiple systems. So we added to our growth and remodeling rule these inflammation terms. I won't go into too much detail, with the exception of saying that what it appears to us is that there's a threshold of stress. And if there's a perturbation stress that's below the threshold, this mechanical process is sufficient. But as soon as that stress exceeds a threshold, then you engage two possible processes. Para-inflammation, if that perturbation or that excess is small, or true inflammation if it's large. And without going through this, we feel that a simple first order equation that essentially relates the stress to the level of inflammation was sufficient to allow us to perform these predictions. So here, what you see, and we simply have one time for this hypertension. Now we've come back later and we have multiple times during the hypertensive period, but here's the first data point. So I'm showing you the first result, not the most recent. And what you see is that the black line is essentially including in this threshold-based inflammation. And it allows us to describe pretty well this observation of the loss of homeostasis, the maladaptive response. And in the light curve, this is when there's no inflammation involved, and this was the 129. So we think that the model gives us the ability to describe these data pretty well. But it's phenomenological. It says you change stress, I'll put a gain in, and that gain tells me how sensitive it is, but it's phenomenological. So a brilliant postdoc in the lab, Linda Irons, said, you know what, we can preserve this constrained mixture theory, but instead of having phenomenological relations for production and removal, let's build cell signaling networks to inform the continuum level model. So we can look at a tissue level, but try to exploit information at a cell level. So here we solve the continuum problem with our constrained mixture theory, which will tell us what is the pressure-induced mechanical stress, both pressure-induced here and flow-induced here, and that these increases or changes in stress will affect through various signaling nodes, and I'm in mechanical engineering, so I won't go this in detail, but there are going to be certain receptors that will have downstream signaling in response to this. And this is what we refer to as mechanotransduction. And some of the key pathways for those who know, Rho-ROCK, which are related to actin-myosin, the mTOR pathway, AKT-mTOR. mTOR is a mechanistic target of rapamycin. It's involved in a growth response. The MAP kinases, which are common, and so-called SMAD pathways downstream of TGF-beta. So outputs can be cell phenotypes, smooth muscle cell phenotype, production of matrix, removal of matrix. We plug this into the model, and what Linda did is she used now a time course sequence of data at multiple times. This is the pressure increase. This is prescribed in the model. And these are the predictions. So the symbols represent data. The solid lines are predictions based on a cell signaling model informing a continuum model. And what you see here is this increase and decrease. This increase and decrease in the biaxial stress consistent with homeostasis. Now, what I didn't tell you is this is the infrarenal abdominal aorta, which is not subject to that dramatic inflammation. So this is truly a homeostatic response without inflammation. And now we're working on cell signaling models to include an inflammation. So there's always the next step to take, and that's for us the next step. And an advantage of these cell signaling models is you can go in and then say, well, what if you used a common antihypertensive medicine? This one's called losartan. It blocks signaling through one particular receptor on these cells. And you can then predict what would happen over time to the different tissue level metrics, and indeed predict what would happen over time for these various signaling molecules, kinases, et cetera. And we're now both using that data to inform the model and using the data to check the model. So let me summarize, and I have a second part of this. There are two vignettes, but the second one is very short. So the tale of two mice. Those two mice clearly had a genetic modifier that they were more C57 than 129. Inflammation really drove that maladaptive response, whereas the mice in the 129 background had this dramatic ability to contract against this exogenous angiotensin II increase in pressure and restore stresses back toward normal. And the key stresses were in the adventitia, that outer layer that was the protective sheath. It was in that layer where we think that the increase in stress, acting on fibroblast, caused them to create cytokines and chemokines to attract the inflammatory cells, which led to that gross maladaptation. Second vignette, which will be much quicker, is unfortunately the most common birth defect or class of defects are called congenital heart defects among live births. And in many of these children, by age 3, they've gone through a series of 3 operations. The third one is called the Fontan completion. And in this third operation, what the surgeon will do is take a polymeric graft and bypass a non-functioning part of the heart to take blood from the so-called inferior vena cava, which is the primary vein that's bringing blood back to our hearts after it's gone to the lower extremities and lower part of the body, and bypass the heart and take that blood directly to the pulmonary artery to then go to the lungs and get oxygen. The good news is this is a life-saving procedure. These children, which in years past would have died prior to age often 6, are now living into their 40s or so. That's a great success. But they still have what's called a Fontan paradox, because we've changed, not we, the community, the surgeons have changed the circulation in a way that it's not optimal. So a surgeon at Nationwide Children's Hospital, Chris Brewer, contacted me, and they were able to reduce this conduit down to the size that would fit in a mouse and actually make it not out of a polymer, but to use a tissue engineered construct. And the way this works is they actually put the polymer in the body, the cells would infiltrate that polymer. As the polymer biodegraded, the cells would start producing matrix. So this sounds like multiple constituents, production and removal. Yeah, we have a theory that maybe we could apply to that. So schematically, here's the scaffold that was built to be implanted in the mouse. And by the way, this diameter is less than 1 millimeter to give you scale. So if you take your ink pen and just draw 1 millimeter, that's smaller than that's the lumen. And this is being implanted in a mouse. What you see here, if you see any bright dots, this under polarized light, the bright dots show you the polymer, the so-called polyglycolic acid. And what you see is the polymer is going away. So conceptually, you start off, the scaffold is all polymer, and at some time, the polymer's gone away. Just like the good and bad news for your aorta, when you lose something, that's bad news, but when you replace it, that's good news. And so fortunately, cells will infiltrate this porous scaffold, and you see the bright dots, that's the polymer, but hopefully you can see red here. And the red is now extracellular matrix, native matrix that's been made by the infiltrating cells, primarily smooth muscle cells and fibroblasts. So ideally, you're losing the polymer, and you design it to degrade on a certain time course, and the cells are producing matrix. So again, this sounds like maybe a constrained mixture might work because we have a polymer and we have collagen, we have other constituents. And so the polymer is not produced, so there's no integral here. But once implanted, it will degrade. So we have a degradation function Q, and we have a stored energy function, which I talked to a few people today, and it's a neo-Hookean type relationship. But the cells will infiltrate and they will produce matrix, which will have a finite half-life and individual properties. So we took the same exact theory, but remember, these are general functions that we now need to specialize for the conditions of interest. And so here is the basal production rate of cells. But remember, they're not there initially. So there's a delay function to account for the fact that it takes time for the cells to get into the scaffold, after which they make matrix. And they make matrix according to this stress-mediated production. And the CP here is the shear modulus of the polymer. And S is the growth remodeling time. So at S equals 0, CP 0 over CP 0, that's 1. 1 minus 1 is 0. So at time 0, the polymer completely stress shields the native cells. So they're not feeling the stress and they're not responding to it. But as that polymer degrades, this CP goes towards 0 and you get a gain and a stress difference. Looks familiar. For the inflammation, we actually tracked the infiltration of inflammatory cells, in this case, macrophages, and prescribed this phenomenologically just to see if we could model what was going on. But the key thing here is you put in a polymeric scaffold that's a foreign body. Our cells recognize that, and the immune cells say we need to respond to that. So the tension stretch curve for the native inferior vena cava looks like this. Common, again, this is an archetype for soft tissue, initially very compliant and then stiffening. This is the polymer. And what we would like to do is compliance match. So we want this tissue engineered graft to evolve in the body from something that looks like this, neo-Hookean, to something that looks like this. So we parameterized our model, and our model description is going to be shown in this dark line, and you'll see some circles here. And at each time, this is at 2 weeks after implantation, this will represent multiple mice. And so what you see is here's the model, a little bit over prediction, but here right in the middle after 6 months. So we're modeling this process of in vivo degradation and, if you will, regenerative medicine in vivo. The clinicians, as we were working towards optimizing the structure, came to us and said, you know, there's a real clinical problem. They felt confident enough, based on some preliminary work, they had FDA approval to implant these in children. These are 3-year-olds. Stakes are high. So this is an angiogram of a scaffold, a tissue engineered graft, and you can see these dotted lines demarcating the two ends. And what you notice is something that's not good. It's a narrowing. We refer to that as stenosis. So I have on the bottom cautious intervention. These are 3-year-old children. So the interventional cardiologist saw that, and if this was a patient, say my age, and that was a coronary artery, that's stenosis, they would go in, either bypass surgery or place a stent, open it up, or angioplasty. So they were concerned, and they went in, they used angioplasty, and this is the outcome. It was very favorable. All 4 of these patients did well, but the FDA said, that's a severe complication, stop the trial. And that's when they had come to us. And I'll never forget the day that we were sitting with them, and my PhD student said, you know, that's very interesting, this narrowing. And he said, our computational model predicts that this graft will narrow. But our model suggests that it would spontaneously return to its normal diameter, which caught the attention of the clinicians. So here's a finite element model using that same growth and remodeling code with the inflammation and mechanics combined. And we're going around in time. And it's a little bit hard to see, but this graft is narrowing and then opening up. And so if you read the numbers, and I can't read it on my screen, it's so faint, it looks distinct there, but you can see the percent narrowing. So it becomes narrow and then it opens back up. This computational result was strong enough that, working with the team at Nationwide Children's, we implanted these grafts in sheep. This is a FDA-approved animal model. And so here you see the implanted graft. The model predicted that this graft would narrow. And there you see it. The model predicted that the graft would spontaneously open up. Now, natural history of a stenosis, say, for example, me, if I have some occlusion in the coronary artery, they don't open up spontaneously. So clinically, if you see a stenosis, it's only going to get worse if it doesn't at least stay the same. So the model predicted this. The LAM study confirmed it. We reparameterized our model based on the LAM data, and you can see the model in the solid line. This is the decrease in diameter. Of course, the wall thickened, and what was happening was the inflammation was driving matrix production. The scaffold was in place. There was only one place for the matrix to go, and that was in, and it encroached on the lumen. But the scaffold would biodegrade, which meant that that collagen was being pushed out by the normal blood pressure. The cells were now feeling the stress, and they started turning over the matrix because the stresses were too low. They were removing matrix. So this was what we call a immunomechanical response. And I'm really happy to tell you that the FDA reapproved the trial based on the computational model. So all of you students, these things can matter. The computational model that then drove a preclinical study. Now, the model suggested that the narrowing was driven primarily by inflammation. So we went in and we took histological samples at 6 weeks where we saw the maximum narrowing. And indeed, looking at the immune cells, that's when we saw, well, this is the wall thickening, and here are the immune cells, and this is, again, a pan-leukocyte marker. I can't read on my screen. Is that a 45 or 68? 68. 68. Okay, so this is the macrophage marker, and the same thing was true for the C45. So again, what the model had predicted was borne out by the experiment. So let me conclude here and tell you that in hindsight, it's been 20 years. Where did 20 years go? But we decided to take a different approach to model what we call growth, change in mass, and remodeling, change in structure, by using a mixture theory for mass balance and a classical linear momentum balance, formulated a single theoretical framework with this constitutive postulate, and spent the next 20 years finding for each individual case, what are the specific constitutive equations for production and removal and properties? Hypertension, aneurysms, you can go around, vasospasm. So this is a neurovascular application, intramural thrombus. When you think about it, thrombus, stroke, heart attack, pulmonary embolism, major cause of death. We modeled this. Aging, which is becoming increasingly important to me. AAAs, inflammation, so et cetera. So each of these dates represents a paper that's been published using a single theory, specialized for the different cases. And when I talk to my students, I say, what's the constituent equation for water? And depending on what they're thinking about, they'll give me one of three answers, and I'll say, we don't have constituent equations for materials, even for water. At room temperature, we might use the Navier-Poisson equations because of viscous fluid. At high temperature, we might use ideal gas law. At low temperatures, we might use an equation for brittle solid. It's the conditions that matter, and in each of these cases, the conditions are slightly different. Some are driven by inflammation, some are not. And so it's the search for these constituent equations that makes all the difference. So with that, I'm going to conclude. I'm going to thank you and simply say that this work, which I get to go around and talk about, really has been a remarkable journey for me in collaborating with individuals. And I've just highlighted here the few who were involved directly in the data that were presented. I probably should have bold-faced some others who were critical along the way, including Alberto Figueroa here at Michigan, and many students and former trainees, which made all of this possible. So, John, I'm going to conclude there. Thank you all very much for your attention.
Thank you very much, Jay, for a really thought-provoking and excellent talk. I'd now like to open the floor for questions. I'm sure there may be quite a bunch, so yeah, we'll start here.
Thank you very much for a very comprehensive model. You've used it sort of. You mentioned the drug efficacy, basically, and tissue design. Just taking it one step further, do you foresee any role of predictive as far as from initial measurements of blood pressure or blood cholesterol, predicting the prognosis of a patient with time based on this type of model?
We do. So that is the hope. And again, it comes back to this comment that one could have a common framework, but different applications which demand different sort of constituent equations or modules. So in the case of the drugs, we think that we need the cell signaling models. And we've shown in a couple cases where, in fact, in this clinical trial, they give losartan, which I mentioned, which is this used as an antihypertensive medication. But they give that drug for 2 weeks at a critical window of time. And I think this model can tell you, what if you go in and block this receptor for this duration? And hopefully be able to catch disease processes at critical periods so that person doesn't have to stay on that drug forever. You prevent that maladaptive response. So the answer is, I think, yes. We do think that we can predict some of these outcomes. We have a new award coming where we're looking at thoracic aortic aneurysms. And we think that this modeling, both the phenomenological and the cell signaling, the coupled model will help us. And I was talking to Krishna earlier, but you know, we don't want to overlook other possibilities. And so the third arm of that study is to use machine learning, but we need a lot of training data for that, and often more data than you can get clinically. So we're going to use these models to generate synthetic data sets because they're pretty reliable to then inform machine learning. So I think that there are multiple applications. It's a great question.
Thank you very much.
Yes, thank you.
Yeah, further questions? Good.
See, cell signaling, is the cell signaling model, is that well characterized or is it something that we have to learn every time?
We need a lot more work. So if you look at a lot of papers in cell biology, you will see these signaling pathways. In fact, you can go to common textbooks and find the MAP kinase pathway, et cetera. But they're not well quantified and certainly not interactive. So the network that we've used came from Linda Irons mining the literature. It was based on 100 different papers in vascular biology. But the network is actually generated by a program called NetFlux that Jeff Saucerman at UVA created. So does it always create the correct network? Not necessarily. So I think that we need to use tools that are available open source, but we do need to mine the literature, and then to validate the model, we have to collect problem-specific data sets. So we're in that process. Now, one thing that surprised us is we've built, this was a homogenized smooth muscle cell fibroblast model because we're interested in the median adventitia. We've almost finished developing an endothelial model, but key on our list is to develop a macrophage model. And there's not as much information available except for macrophages that are in the abdomen. So I think that's going to require a lot of sort of basic science experiments to really nail that down. So in summary, we can glean a lot from the literature. We can build these models, we can test them, we can refine them. But I think very specific data sets will be needed to really say, this is a reliable model for this special case. And that's our goal. Yes.
Just following on the conversation here, there are other like common disease, atherosclerosis, involving transformation, differentiation of immune cells. I'm wondering your continuity, like the continuum model, growth and modeling in the future, you might be able to take into account some additional disease driving mechanisms. Comment on that.
Absolutely. And I think your atherosclerosis is a leading cause of death, a critical problem. A PhD student of mine years ago, working with a different group at UVA, Jeff Saucerman's at UVA, Shane Pierce-Kotler, developed a combined agent-based model and a constrained mixture model to look at athero. But I don't know about here in Ann Arbor, but in New Haven, there are only 24 hours in our day. And so we can't address everything. So we've really focused more on aortopathies in the last really decade. And atherosclerosis is not a major problem of the aorta. It's coronary arteries, carotid arteries, renal arteries. So we haven't undertaken that, but I hope that someone will pick up that because it's a very important problem. I do think that this approach would work. It's complicated, but it's defined by all of these different interactions amongst multiple cell types and different constituents and laying down new collagen in the fibrous cap and necrotic core and all of this. I think that one could use this approach. We just have not done that. And to be honest, I don't see that happening in the next 5 years. We've got our hands full with another, quite a few different projects. Yes.
Do you use spatial transcriptomics data at all?
Not yet, but that also is on our list. So what I showed you in the hypertension study was bulk sequencing. We've obviously done single cell sequencing to look at these cell-cell interactions in multiple cases. But as you correctly observed, if something's happening in the media that's different than happening in the adventitia, single cell should help sort that out because fibroblasts are in one layer, but spatial transcriptomics will play a role, as will proteomics. So we have to remember that's just the transcript. It's not telling us what's being translated and what the actual protein is. And it's the protein that has the half-life that we're most interested in. So I think both of those techniques will need to be added. Yes.
So this was mostly being used in large arteries. I'm kind of interested if it could be applicable in small arteries and maybe even in something where you could predict like rarefaction.
Yes. The answer is yes. I do believe that it could, and we have another project that's just starting. Again, there's just too many. But on the pulmonary side, looking at pulmonary vascular resistance, which is a key issue in these children with congenital heart defects. And I do think that the model could be extended, but I think that's where you need to develop a multi-fidelity approach where maybe some lump parameter modeling might augment this. It's just like you don't have to solve the Navier-Stokes equations everywhere. You can solve them in domains of interest and then couple them to reduced order models. I think that will be necessary. But as far as the general idea, yes, particularly if you get cell level information, like what is the endothelium doing, which is different from the smooth muscle cell. We know the smooth muscle cells are phenotypically different in resistance beds versus large vessels. So in principle, yes. I don't know of anybody that's doing that, so there's an opportunity. Yes.
Not related to the previous question, but and I know you said you're very busy with a lot of different projects, but have you guys considered like looking at families with kind of hereditary like aneurysm diseases where there are like known kind of genetic defects which have like kind of known pathways? Then have you thought about kind of like modeling what would happen if you kind of like adjusted certain things to like target certain kind of like just like nodes in that pathway and like see like what would happen if you, because you talked about like losartan, and I know that's like a pretty common like anti-aneurysm drug right now.
Both common and controversial.
Yes.
So you must know something about thoracic aortopathy. So the answers to your questions, which were maybe three, are the answer is yes, yes, and yes. So we have Marfan mice in the lab. We have TGF-beta mutant mice in the lab. We work, I don't know if you know the name, Diana Milovitz at University of Texas, Houston is one of the leading geneticists who discovered the ACTA2 mutation in humans, among others. So yes, we are working on that. And that is one of the key things that we're looking at, are genetically predisposed thoracic aneurysms and dissections.
So I was struck by something you said kind of in passing, but it was that cells pre-stress collagen during CM deposition. So they pre-stress it to match the stress in the environment that they're in. So they'll stretch that triple helix to different amounts. Wow.
We conjectured that based on the model. So there's one parameter in the model and you have collagen fiber families in all these different directions. But if you put this one parameter and set it to 1, which says that it's all chemistry, the cell just secretes these molecules and they self-assemble like they do in a test tube. The model said that the artery would have unbounded growth. And if you think about it, what you're doing is you're replacing stressed material on the wall. What happens if you replace stressed material with an unstressed material? The answer is it will deform until it's stressed. It's like that door frame. If you took out that door frame and put in one that was 6 inches lower, what would happen? The wall would probably come down until it met that support, right? So if you want to replace it without a change, you put it in at the same stress. And we developed a theory that we called a theory of mechanobiological stability. So actually took, just as in mechanics, you have equilibrium and you have stability analyses, we have a mechanobiological equilibrium and a mechanobiological stability. And that theory suggests that you have to have pre-stressed material. So then we went in the literature and asked, is that possible? And Carl Cadler in the UK has data that shows in neonatal fibroblast that they will produce collagen in culture and they will organize that collagen. But if you block actin, they will produce the same amount of collagen, but it's not organized. That those cells are actually, and if you read Albert's, seminal book on molecular biology and read the section on fibroblasts and collagen, they knew that. They said that these cells, I forget the exact, but they crawl over and they work on the matrix in order to fashion it. So yes, we do believe the cells are pre-stressing this material. And with regard to the elastin, it is unique in that it's cross-linked early in development, has a very long turnover rate, half-life in the order of 50 years, which means that the elastin you're born with is what you'll die with, pretty much, with some loss. You'll make it to about 100, and that's about right. So with that exception, all of these matrix materials are being turned over, and we think they're being pre-stressed. But even elastin is pre-stressed in the developmental period. And Bob Meekum, who's an expert on vascular elastin at WashU, has these videos that actually show cells which are producing the elastin or the tropoelastin, pulling on it, stretching it out. And remarkably, we had predicted that the pre-stretch should be about 50%, stretch of 1.5. And those videos, you watch them and it's about 50%. So yeah, these cells, it's not just the chemistry. They don't just secrete the material into extracellular space and say self-assemble. They work on it and they organize it and they direct it. And in so doing, they put a pre-stress in it. It's actually critical for the stability of the vessel. Yeah.
Just wondering, in a lot of this work, I guess, so we have the models, we have these models which were postulated in a sort of phenomenological sense, but for good
Absolutely.
reason, right? There was an understanding, as with any phenomenological model. I was thinking all along that there's probably a lot of room for cell mechanics work or cell biophysics, maybe not just mechanics, but definitely, right? Inflammation, especially when you spoke of a macrophage model and so forth. And what you now just described about the stress, about the cells sensing the state of stress and depositing collagen at that state of stress suggests that there's a great deal more to be understood there. It also brings us up to this old one, we were talking about controversies earlier in the morning in this business about the one where the cells sense strain or stress, and how can they possibly sense stress, right? The old fights, right? So what's about
Yeah, so good point. So the first one is we have a tissue level model, you could say tissue organ level. We have a cell signaling model that's intracellular. We skipped over the cells. And I think that level should be put in. So the answer to that is yes. With regard to what do they sense? I actually wrote a paper. I'd been to this tissue engineering meeting that there was this cell sense stress, cell sense strain, and it was this back and forth. And I just got up and said, neither. Said, I don't know of any cell that's taken my continuum mechanics class and knows what a linear transformation is that takes an outward unit normal vector and turns it into a traction vector, or a tensor that takes a differential position vector and turns it into another one. So I think that stress and strain are these wonderful concepts that we have sort of intellectualized and we use and they're reliable, but I'm not convinced that they exist, period, and certainly not at a cell level. So what does the cell sense? Probably conformational changes in molecules, signaling molecules of importance. But as any case, when we walk into a laboratory and we want to measure something, we often can't measure what we want to. It's just like the old temperature. You didn't measure temperature, you measured how much of mercury, column moved up, and you put a scale behind it so you could say, well, it's whatever the temperature is. So I think that these calibrations are really useful. And I think that stress on the vascular side is a really nice surrogate marker, continuum marker that we can calculate and through a calibration relate to what the cell's doing. It doesn't mean it's responding to that, but it means that we have a correlation and it's useful and it's predictive. So the biophysicists will need to continue to look at, and I've got colleagues at Yale, Martin Schwartz among them, doing really clever work at looking at the mechanotransduction and which parts of which molecule is contributing to that signal. But yeah, to answer your question, I don't think they sense stress or strain, and why should we argue about it? But what they do is they respond to mechanical stimulus, and we can get really good correlations between changes in stress in the vasculature and their response. And at Yale, I don't know about here at Michigan, but when students come into my intro mechanics course, I tell them, if you're looking for truth, go to the divinity school or the philosophy department. If you're looking for utility, this is the right place, because we're really trying to find relationships that are useful that can help predict what's going to happen to a vascular graft when put in a 3-year-old child. It didn't matter that I didn't know whether it was sensing stress or strain. The model was able to predict that natural history and hopefully change outcomes for those patients. That's utility. And I think that that's a modest thing to search for as a biomedical engineer.
I don't think we could close on a better one. So thank you, Jim. Appreciate it.